Hello and welcome. In this video, we will discuss randomization in clinical trials. If you are interested in conducting clinical research, then you must make a team and get in touch with the biostatistician in your institution. Proper planning will go a long way in showing validity of your findings. So in this video, we will be discussing randomization, and we will find out what is the meaning of randomization, why is it important, in which trials can we randomize and in which we cannot, what is the process followed in randomization. So moving on to the meaning and the benefit of randomization. Randomization is a critical process in research, particularly in clinical trials, to ensure that study participants are assigned to different treatment groups or conditions in an unbiased and unpredictable manner. This helps eliminate selection bias and ensures that the groups are comparable, enabling researchers to draw valid conclusions about the effects of a treatment or intervention. Studies can we apply randomization? So this can be applied in RCTs or randomized controlled trials. These are often considered the gold standard for evaluating the effectiveness of medical treatments or interventions. Another kind of study is randomized clinical trials. So actually, randomized controlled trials and randomized clinical trials are very much similar to each other, and sometimes the term RCT is used interchangeably, but the only difference is that randomized clinical trials may not necessarily be double-blinded, meaning that the participants and or the researchers may be aware of the group assignments. A third kind of study in which we can apply randomization is the cluster randomized trial. In some cases, it may not be feasible to randomize individual participants. Instead, randomization is performed at the level of groups or clusters such as hospitals, schools, or communities. And this is often used in public health and community-based interventions. I have given a reference in which we performed a community-level intervention for preeclampsia in Pakistan, and this was a cluster randomized controlled trial. If any of you are interested, you can go and look it up. Blinded randomized controlled trials are research studies which have been designed to reduce bias by withholding specific information from either the participants, the investigators, or even both. The level of blinding can vary, leading to different terms like double blind and triple blind. In a blinded study where it is single blinded, typically the participants are unaware of the group to which they have been assigned. So the participants sign a consent form for inclusion in the study, but they are not aware whether they are in the control group or they are in the intervention group. This blinding prevents participants from altering their behavior or reporting outcomes in a biased manner based on their knowledge of their group assignment. However, the investigators and the researchers are aware of the group assignments. They collect and analyze the data, which introduces the possibility of observer bias. In a double-blind study, both the participants and the investigators or the researchers are unaware of the group assignment. This design helps eliminate both participant bias and observer bias. To achieve this, a third party or a computer system may be responsible for generating and maintaining the randomization code, ensuring that the allocation is concealed from everyone involved in the study. In a triple-blind study, the term implies that the three groups of individuals who are involved primarily remain unaware of the group assignment. And these three groups typically include the participants on whom the study is being conducted, the investigators who have planned the study, and who have done the randomization of the groups, and often the data analysts or the statisticians responsible for data analysis. So in other words, there will be a code assigned to each group, and once the results are made available, then you will uncode them and you will find out which group did better. Now let us look at the kind of studies where randomization cannot be applied. So observational studies are those in which the researchers collect and observe data on participants without intervening or assigning specific treatments. These studies are primarily used to establish associations or correlations between variables but do not involve randomizations. Another one is case reports and case series. They involve detailed description of individual patients or a small group of patients who have a rare or unusual condition or response to treatment. There is no randomization in these types of studies as they primarily serve to document unique clinical experiences. Descriptive studies aim to characterize the prevalence, incidence, or natural history of a particular disease or condition. They also do not involve randomization. Ecological studies examine the relationships between exposures and outcomes at a population or a group level rather than an individual level. Qualitative research methods such as interviews, focus group discussions, and content analysis aim to gain insights into the experiences, the beliefs, and the perspectives of participants. There is no randomization involved as the focus is on understanding subjective experiences. Other kinds of studies that can be performed and where randomization cannot be done include retrospective studies, in which data is collected retrospectively and therefore randomization is not possible. In addition to that, longitudinal studies, while some longitudinal studies may involve randomization, but many do not. Longitudinal studies follow participants over an extended period to examine changes in health or other outcomes over time. Randomization may be challenging in such studies due to the extended duration and the ethical considerations. Post-marketing surveillance are the kind of studies which monitor the safety and side effects of medications or interventions after they have been approved for use. Randomization is not typically used in these studies because patients have already been prescribed the treatment. Health service research means audits, and it also means interventions for improving health policy, and randomization is not applicable on practical when studying healthcare system structures and policies. So moving on, explain in a very, very simple way the concept of randomization. Let us imagine that you want to do a study on endometriosis patients. Now, supposing that you have 6 cases of these endometriosis and you are aware of the names of these 6 patients, but you want to test a drug called the miracle drug, and you want to test it on 3 patients and keep 3 patients as a control. So how are you going to now decide which 3 patients are going to be the study patients and which 3 patients are going to be the control patients? So in the study patients, you will give the intervention of the miracle drug, whereas in the control group, you will give no drugs or just the drugs that they are always already taking. So the concept of randomization is similar to that of throwing a dice. So when you throw the dice, then you can either get a number 1 or a 2, 3, 4, 5, or 6. So supposing that you have a list of your patients, this is the list of the patients, and you throw the dice and you get the number 6. That means that number 6 is now going to be the intervention group. Then you throw the dice again and you get the number 4. So now the 4th patient on the list is going to be has now been randomly assigned to the group in which the intervention will take place, and she will be given the miracle drug. The third time you throw the dice again, you get the number 1. So now randomly out of these 6 patients, you have selected that 1, 4, and 6 is going to get the miracle drug, and 2, 3, and 5 will get a placebo, or will be having their normal treatment. In this way, you have randomly assigned the intervention group. So now this process of randomization, where you throw a dice, can be done by other methods, like for example by the computer, in which you can ask the computer to generate numbers from 1 to 100, or ask the random numbers from 1 to 100, or 1 to 1000, or 1 to 10,000, or 1 to 20,000. So when you have very large numbers, then you can't keep throwing the dice, or 2 dice, or 3 dice, because whenever you throw a dice, there is a certain limitation that what numbers you can get. So now that you have hopefully understood the concept of randomization, let's move forward into the process of randomization that we use when we are doing a random assignation of large groups, large study populations. So the first thing that you do is you define the study population. So how do you define the study population? You first of all select what is going to be the eligibility criteria, who are the patients whom you will allow to be entered into the study, and you will have fixed criteria, standardized criteria, which may include age, gender, medical history, or even other relevant characteristics. Once you have defined the study population, so the next step of the process is to generate a randomization plan. And the randomization plan is typically created using either computer software, or you can use tables in which random numbers have been written down. The goal is to produce a list of unique random assignations or assignments, such as treatment or control, that will be used for each participant. This list is often referred to as a randomization schedule. Now, to maintain the integrity of the randomization process and to prevent selection bias, allocation concealment is crucial. This means that those who are responsible for assigning the participants to groups should not have knowledge of the upcoming assignments. This is often done in those studies where there is a placebo and there's a drug. The placebo and the drug look exactly the same and are in exactly the same kind of package, and they are put inside the envelope, and in the envelope inside there has a number, and that is the number that is assigned to that particular package of drug, and that determines whether this patient is getting the placebo and is in the control group as compared to the other patient who may be in the intervention group and is going to get the drug. So the common method for concealment includes sequentially numbered opaque sealed envelopes, and so that you can't look inside or you can't even against the light, or a centralized secure computer generated randomization numbers system. So this is another way. The centralized secure computer generated randomization number is another way in which you will enter the details of the patient, that this is her name, and this is the date at which she has come, and this is the time at which she has come. And then once you have entered it and you have submitted the form about the patient, the computer will send you a number, and this number is going to be assigned to that patient, and then this patient will then be either in the control group or in the intervention group. So basically, this is not a very simple and straightforward process, because it has to be written down in great detail and great fine tuning, so that the randomization process is kept concealed and not opened up to the people who are giving the drug to the patient, which is very, very important, because that can introduce bias. So researchers began enrolling participants into the study one by one or in batches, depending on the study design. Each participant is assigned a unique identification number, and this identification number will be carried on till the end of the study, at which time the results will be made available by opening the code. Using the randomization schedule, the research teams assign participants to their respective groups. For example, if the study has 2 groups, one might be in the treatment group and the other in the control group. The allocation is typically done sequentially, ensuring that neither the researchers nor the participants can predict the next assignment. Tracking and record keeping. Accurate record keeping is essential. The randomization process should be documented thoroughly, including the date of randomization, the assigned group, and the unique identification number of the participant, and this ensures transparency and traceability. Throughout the trial, it's important to regularly monitor and assess the randomization process to ensure that it is proceeding as planned. Any discrepancies, errors, or issues should be addressed promptly. In some cases, researchers might need to unblind the randomization if an emergency situation or an adverse event arises. But this should be done with caution and reported in the trial's documentation. After the data collection is completed, statistical analysis is conducted to compare the outcomes of the different groups. Reporting means when you are either making a report or publishing. The randomization process, including any details about the randomization plan, allocation concealment, and unblinding, should be clearly described in the final study report or publication. This transparency is essential for peer review and the credibility of the research findings. The choice of randomization method should consider the trial's specific design, objectives, and potential sources of bias, as well as the overall goals of the research. The biostatistician, the epidemiologist, and the senior researchers should be involved to fine-tune the randomization process. Therefore, randomization is the cornerstone of evidence-based medicine, allowing researchers to draw valid conclusions about the efficacy, the effectiveness, and the safety of medical interventions. With this, we come to the end of our video. If you like the video, then please subscribe, like, share, comment, and press the bell icon for information about future videos. Thank you and goodbye.